All right, guys. So we're gonna kick back up here with translation and transcription. Now we talked a little bit about translation and what it was. We didn't really give it a name. We just talked a little bit about DNA. So remember, DNA is a double helix, ATGC, right? And it's really how we store information, right? The next part, when we make RNA, is we're going to have something that is readable and ultimately make proteins, which is something that we can actually use. Quick refresher on RNA, ribonucleic acid, similar to DNA, except it is single-stranded, not double-stranded. Its sugar base is ribose, not deoxyribose. We use uracil instead of thymine. And we have several types. We have mRNA, rRNA, and tRNA, which we will talk about each independently. So, the whole point of transcription is to make our guy down here, which is our messenger RNA. Okay. So, just like we had DNA, when we did DNA replication, we had an enzyme called DNA polymerase, which added those nucleotides in. All right. So, here we have RNA polymerase. Very similar. What it'll do is it'll come in and it'll unzip this section. It'll cut this DNA, break those hydrogen bonds, and then it's going to latch on here and go down the line. In this case, we're reading this template right here. It's going to go from 3' to 5', and we will make our mRNA. So once again, during transcription, the RNA polymerase binds to the DNA, separates the strands, goes down. We put our nucleotides in according to our DNA template strand, and we end up with an RNA section. Okay, so just a big kind of overview here. We have don't worry about introns and extrons. We're not really doing that at this level. So we have our DNA. We turn it and we make it into RNA. Then we're going to feed our mRNA through a rRNA. All right, and as it goes through, we will translate, all right, into these amino acids using tRNA, codon, anticodons, and all that good stuff. So, DNA polymerase binds to the promoter site, and we end up with our RNA section. So here's a little example. Oh, here's transcription. All right, so we have our. I'm not going to really can't really stop the little animation, but you can see here our RNA promoter comes on, latches on, goes down, and makes our piece of mRNA. All right, so a little bit of a refresher. Proteins are made up of amino acids. All right, our amino acids right here are carried by our tRNA. Remember, T stands for transfer, so they're going to transfer in the RNA. They're going to transfer in the amino acids. There are 20 amino acids, and we read our mRNA section in sections of 3, which is called a codon. So our mRNA holds the codon. Our T, and we'll see this in the next slide, I do believe, tRNA is going to have what we call an anticodon. All right, and there's some just examples, and we'll talk a little bit more about that in class and in this lecture. So this is what we use for when we try to decode our sections, and this is an mRNA chart. So you will use your mRNA section to come up with the appropriate amino acid that you will be using. Once again, I'll never ask you to memorize this chart or anything like that. You'll have this and the gene wheel when it comes to a test or a quiz, so do not spend any time memorizing this. What you need to understand, for example, if I had AUG, okay, you use this is your first position, so A, second position, U, third position, G, so that means I am here. All right, methylene, and that's kind of a start codon. That's why I did that one. Okay, so as another example, let's do, these are totally random, just coming up with these off the top of my head. U, C, C, which would be serine. Okay? So, as we talked about, our tRNA is going to bring over and have the appropriate amino acid. We have our structure. Know this structure. Know what it kind of looks like. General shape. Not perfectly, but general shape. When we also have our anticodon up here. Okay? This is where we will pair with the mRNA strand. And then our rRNA is kind of the example I always talk about, is a staple or bond each individual amino acid together to a polypeptide chain, aka a protein. Okay, so, and I'll kind of draw this in a minute. The cell used the information from mRNA to produce the proteins. The mRNA attaches to the ribosome. It moves to each codon down the way, and the ribosome will bind them together as the tRNA brings in the amino acids. Okay, and then, like I said, eventually you end up with a chain. That chain will fold up into a structural and a beneficial protein. A long chain of amino acids is great, but it's not really functional. So we fold it, and then we have something we can use. Okay, so here's a little example. Remember, this ribosome would be attached to the rough ER. The rough endoplasmic reticulum, that's what makes it look rough, is it's studded with a bunch of rRNAs. Comes in, it joins in. All right, we have our tRNAs right here. They have the anticodon portion right here and our amino acid right there. They come down, they bind, all right, and you can see from our little chain here, we're just growing this amino acid chain until something we can actually use. Okay, and this is called the central dogma of biology. DNA goes to RNA. All right, so back to our drawing. Okay, so A, U, G, C, C, A, U, U, C, A, G. Now I came up with these letters totally at random, other than this one, because this is our methylene, which is our start codon. So we're going to read this in sections of 3. Okay. Now, there are 3 binding sites in a rRNA section. Oop. Okay. So 1 binding site, 2 binding sites, 3 binding sites. And the mRNA is going to be pushed through in this manner. So we're going to have a tRNA. Okay. At this point, I'm going to write down the accurate anticodon, so it would be UAC. All right, that is attached to our tRNA molecule. All right, we have our amino acid out here, which is methylene. And then we're going to have another one come down, and we'll have GGU. That's our anticodon. Boop. I know you'll love my sweet drawings. Okay. Then we have something, I don't know off the top of my head. All right, so we have another amino acid, and then our rRNA molecule is going to form a bond between there. And then it's going to shift down, and then we'll add in another one, UAA. There's our anticodon. Here's our piece of tRNA. And then they're going to bond them together again. And like I said, as we go through, so the next one, as it would go through, as it moves down, it goes this way. This goes this way. We would have another, this bonding site would empty off. Our tRNA goes out. It moves along. And then this bonding site right here is now, whoops, this bonding site now is here. So if it was there, we would have GUC. All right, anticodon. A sweet drawing of a tRNA. And then we have our amino acid here ready to get added to this chain. All right, so that's a brief overview of translation. We'll start with transcription from DNA to RNA, and then translation from RNA to something that we really need, something that we use in our body, which would be proteins.